Good day, everybody. My name is Kieran Jacoby, and I'm currently a master's student at the University of Kwa-Zulu Natal at the Mandela School of Medicine. My presentation today will be on investigating the methylome of the clinical strains of Mycobacterium tuberculosis complex. Mycobacterium tuberculosis, also known as MTB, is the causative agent of the respiratory disease tuberculosis, which is also known as TB. This disease has raised health concerns worldwide due to the estimated 1.6 million deaths, as well as 10.6 million new cases in 2021 alone, as estimated by the World Health Organization. Additionally, TB is the leading cause of death in individuals with HIV due to their immunocompromised status. The high incidence rate of tuberculosis worldwide has been attributed to both the immunocompromised population as well as the recent emergence of multi-drug and extensively drug-resistant strains. These strains have thought to have come about due to the mismanagement of drug treatments, as well as person-to-person transmission, resulting in the acquisition of drug-resistant genes. There's been great concern raised because of these strains due to the fact that they are able to resist our first line of defense in terms of drug treatment, resulting in us having to resort to less effective drug treatment methods, which also sometimes they are resistant to as well. So this has greatly increased our demand for the development of novel drug targets and treatment methods in order to adequately combat this ever-growing threat of resistant strains. In order to do this, we need to better understand the different mechanisms at play in tuberculosis so that we can uncover novel targets for the development of new treatments. The Mycobacterium species that are the causative agents of tuberculosis in both humans and animals are grouped within the family known as the Mycobacterium tuberculosis complex. This complex consists of a variety of different species, which are categorized into 9 distinct phylogenetic lineages. Originally, we thought there were only 7 lineages. However, recent studies into the identification have shown that there are now 9. The interesting thing about the species within this complex is that they show significant genetic similarity between one another, even species from different lineages, as they show a greater than 99% identity at the genetic level, and it is theorized that they all evolved from a common ancestor. Despite this great similarity, they show a variety of distinct phenotypes, diverse biological functions, as well as a wide spectrum of different hosts, with even species from the same lineage. Therefore, we need to try and understand where this diversity comes from, if not at the genetic level. Recent studies have attributed these differences to transcriptional regulation of gene expression, resulting in these different phenotypes. Additionally, there has been some evidence to show that this heterogeneity is associated with epigenetic influence and inheritance, with the most common epigenetic regulator being methylation in MTB. Therefore, we are looking into the effects that DNA methylation has and the role that it plays in the regulation of the MTB genome. DNA methylation is referred to as an epigenetic mechanism due to its ability to regulate gene expression in response to environmental stresses. The purpose of this mechanism is to allow an organism to better adapt to its environment to increase its survivability. The mechanism of methylation involves enzymes known as methyltransferases, which bind methyl groups to specific sites on the genome. These binding sites with their bound methyl groups alters the binding potential of the transcriptional factors associated or responsible for gene expression. This can result in either an upregulation or downregulation of the respective genes. This kind of methylation-mediated alteration has been shown to be responsible for some of the differences we see in lineage-specific strains, or lineage-specific differences we see between strains in the MTB complex. Recent studies using the SMART platform have shown or identified 3 methyltransferases, being MAM-A, MAM-B, and HSDM. And all 3 of these have been shown to play a significant role in regulating different parts of the MTB genome. For example, MAM-A has been shown to regulate genes in response to hypoxic conditions, which could allow the organism to better persist in this stressful environment. Additionally, HSDM has shown to methylate drug transporter genes as well as drug target genes, which could mean that it is responsible or plays a role in the drug resistance within MTB. An additional example, within RV1523 MTAs, it has been shown to play a role in the fatty acid biosynthesis, metabolism of different nutrient sources, as well as confer resistance. Therefore, it is important that we understand this mechanism correctly so that we can better have a grasp on how important it is to MTB as a whole. This could open a variety of different novel drug targets or pathways for the development of new treatments to better combat the rising resistant strains of MTB. Aims and objectives for my project are as follows. Initially, we wanted to obtain whole genome sequences from our clinical strains of MTB, and then we wanted to go forth and analyze and profile the methylome of each of these strains so that we could categorize the epigenomic differences as a result of cholesterol-rich environments. So our objectives for this, we first culture our MTB in both 7H9 Middlebrook media, as well as a cholesterol-rich minimal media or starvation media. We went on to do growth curve analysis, and then we isolated our DNA from the strains that were grown in both 7H9 and this cholesterol media. We continued to use the PacBio Smart sequencing platform so that we could get whole genome sequences as well as have the annotated methylation information alongside that, alongside the whole genome sequences. We used the SmartLink system to get epigenomic characterization, and then onwards from that, we're going to try to identify the lineage-specific methylome-associated mutations and get a complete epigenomic characterization. During the life cycle of MTB, they are heavily dependent upon host-derived nutrients for their growth and survival. During the replication phase of MTB, they are present in cholesterol-rich environments such as granuloma and macrophages. Therefore, they need to be able to adequately metabolize these cholesterols to derive the required nutrients and energy. From this, we can conclude that cholesterol plays an important role in the life cycle of MTB. Therefore, the mechanisms of regulation should be better understood to allow us to develop potentially new targets for treatment. Therefore, for our growth curve analysis, we grew our strains in both 7H9 and cholesterol-rich minimal media. And from this analysis, we were able to reveal the fast growers, which were strains 85, 88, 92, and the slow growers, which were strains 91, 94, and 87. The differences we observe in the growth between these strains could be as a result of an upregulation or a downregulation of cholesterol metabolism-associated genes, which would make them either worse or better suited to the environment, and as they are able to better utilize these cholesterols. However, the strange thing that can be observed here is that strains from the same lineages show very great variance in their growth. As an example, strain 92 is a fast grower from lineage 4. However, strain 94 from lineage 4 is a slow grower. So, therefore, from this, we can show that there's an altered regulation in the cholesterol-associated genes, and we can conclude that MTB exhibits strain-specific characteristics, not lineage-specific. Once we had completed our growth curve analysis, we moved on to sequencing and characterizing our MTB DNA. Initially, we grew all of our clinical strains in both 7H9 and cholesterol-rich minimal media. We spun them down, extracted them, and sequenced them using the PacBio Smart Sequencing platform, as it allowed us to gain insight on both the genetic information and the methylome profiles for each of the strains. We then annotated the output of these sequencing using the Diastamo analysis platform, which is a very powerful platform as it allows us to gain insight on the different regions that are methylated by each individual motifs from these different strains. So with that in mind, the example on the left-hand side is a singular motif that was present when H37RV was grown in cholesterol media. This was an additional methylation motif that was only present when grown in cholesterol and not present when it was grown within 7H9, which means this is going to potentially provide the genetic regulation required to metabolize these genes. However, that will be determined with further analysis. So to better understand this image on the left-hand side, the red indicates genes that were significantly methylated by this motif, and they were designated a z-score of greater than or equal to 2. While the blue zones indicate a significantly unmethylated region and is designated a negative or less than or equal to -2 z-score. So for this motif specifically, some of the regions that were methylated, significantly methylated or unmethylated by this motif were as follows. RV3632, for example, plays a significant role in the metabolism of nitrogen, which is an essential component within the life cycle of MTB. And further examples being for the lineage H strain that we sequenced, an additional multiple different motifs were present, not just a singular one of which including the GTAY-NNNN-ATC motif, which was responsible for methylating the FAD-D2 gene, which is responsible for parasitamide resistance, which is a crucial resistance gene for many of the multi-drug resistant strains, and it's important that we understand this mechanism so that we could potentially better fight these resistance. Therefore, the presence of particular motifs when grown in cholesterol media rather than in 7H9 indicates that there are distinct epigenetic regulations taking place between the different strains when grown in different media. Therefore, this kind of information could allow us to gain a better insight into novel treatment development, as it could shed light onto new targets as we better understand the crucial mechanisms involved in the life cycle of MTB. A continuation of the characterization of DNA, and in addition to the Diastamo analysis outputs in the form of the images as seen in the previous slide, we are given the raw output of all the different genes that are affected as a result of the presence of these motifs. As can be seen in the table on the left, we can see the gene names and the z-scores. The z-scores being greater than 2, meaning they are significantly methylated. However, there will be other genes that are less than -2, which shows they were significantly unmethylated. This data allows us to show that there has been some form of methylation, and there's a potential that there will be an alteration in the expression of these associated genes. However, we cannot confer whether it's an upregulation or downregulation. As Diastamo does not provide that kind of information, we would need further protein analysis to show the different levels of gene expression. However, from this, we can understand some of the processes that could potentially be altered or upregulated as a result of the epigenetic alterations and motifs present. Using our results from the diastomo analysis, we ran them through David, which is a gene ontology analyzer. From this, we were able to determine the biological processes of the genes that were enriched by the respective motifs. This allows us to potentially observe the epigenetic differences as a result of the differences in environment, being cholesterol, as well as the presence of these new motifs. The media-exclusive motifs are interesting because they have very similar functions, however different distributions, and they have distinct new functions which we somewhat expected to see. For example, in the image on the left, which was H37RB growing cholesterol, its exclusive motif showed lipid metabolism, which could allow it to have better utilized the cholesterol as its main nutrient source. Additionally, we see a significant increase in the number of genes for things like cell wall, cytosol, and ATP binding, which could be essential for activities such as replication, which is what we expect to see due to the fact that replication usually takes place in high cholesterol environments. However, on the image on the right, we do not see any of the lipid metabolism, which makes sense because cholesterols were not present in our 7H9. However, we still did see a lower frequency of genes associated with cell wall and cytosol, and a variety of other different genes. Therefore, from this, we can start seeing some of the potential differences as a result of epigenetic changes and direct functional differences due to it. We've come to the end of my presentation. Thank you very much for watching. Before we end, I would like to provide a few acknowledgements. Firstly, to the Research Foundation of EDCTP program, who supported my research, who funded my research, as well as the scholarship that I obtained from the Health Science Postgraduate Scholarship Program. Thank you.